Coral reefs, the most diverse biological communities in the sea. Often called cities of coral, these enormous masses of calcium carbonate are the most productive ecosystems on the face of the earth. For the past 500 million years, constant warm temperatures around the equatorial belt have provided ideal conditions for the gradual evolution of this dramatic diversity of plant and animal life. Coral reefs are of growing importance, for they are among the last frontiers of natural history for scientific discovery. If we can learn how these complex and highly productive underwater communities function, perhaps we can find ways to help our human societies survive.
We sail out of Washington, we sail the 7 seas for coastal research and turf ecology. The reefs near the equator, the fjords of old Iceland, the marshes of America, the place just off Japan. Roll on Marsus, Marsus resolute. There ain't no ocean deep enough or wave we cannot shoot. Our captain will lead us through the calms and windy storms. The albatross, our loyal friend, will fly where we roam.
There,
A couple more degrees because we've wandered in. How about sitting right here?
Scientists of the Smithsonian Institution's Marine Systems Laboratory are embarking on a 4-month expedition to study coral reefs in the Caribbean basin. They will travel to remote areas never before known to science. It will be a unique operation. The scientific team also functions as the ship's crew. 12 scientists will sail their floating laboratory on a 5,000-mile journey from the Potomac River to the waters of the Western Caribbean and home. Cape Henry Lighthouse at the mouth of Chesapeake Bay signals the end of land and the beginning of the ocean voyage. Now, as sailors, the scientists must contend with rough weather, constant motion, and the adverse effects of salt water everywhere. Before radar, Loran, and other electronic inventions, traveling the waters off the Carolina coasts was a hazardous adventure at best. And Walter Ady, the lab's director and ship's captain, knows that the Caribbean waters won't be much better.
On his first trip of exploration, Columbus sank one of his vessels on a Haitian reef. And throughout the 16th century, enough Spanish galleons were lost in the Caribbean to make treasure hunting for Spanish gold one of the oldest industries in the area.
Serrana Bank, a remote, uninhabited atoll in southwest Caribbean waters.
Looks like it's near the surface over there, and yet by the time it gets over here...
This is the first of many previously unstudied reefs the scientific team will visit.
What is the depth? Tell them to give us that.
With reef structure living just below the water's surface, they must use caution to maneuver this deep draft vessel through the narrow passage to safe anchorage. The Smithsonian Albatross is an important member of the team. This historic aircraft is one of only a few large amphibians still in operation today.
You should be able to see.
It delivers people and equipment to remote, inaccessible areas and allows the expedition to study many more reefs than would otherwise be possible. Now now the sailors again become scientists and prepare for the tasks that lie ahead. There will be long, wet periods of operating from small boats, handling delicate electronic and mechanical equipment, and many hours working underwater, studying these fascinating coral communities.
Reefs in particular are very complex communities. They're very old communities. There are many, many reasons to simply be curious. Why does it have so many species, for example? How is it that they're able to be so productive? These are all academic questions. The need to know how something particularly as exciting as a reef works. If you take a long, hard look at the best, well-populated typical Caribbean reefs, you see that only a small part of the surface is occupied by living coral, 15 or 20% at the most. The rest of that surface is carbonate that is generally occupied by algal turf, which is the lawn, the food for most of the reef organisms. The base of the plants are embedded in the porous reef structure, and as fast as they're eaten off, the plants just grow right back again and go on with their business of life. Most biologists would say, and this has only been accepted in the last 5 or 8 years, yes, coral reefs are the most productive ecosystems on the face of the earth. They would also say they're very peculiar in the sense that while they're the most productive, they live in the seas with the least nutrients.
One of the first things they do is set up shop at the reef's crest. For the next week, this will be home for many members of the team. Working from platforms, the scientists measure current flow and oxygen levels as the water enters and exits the reef crest. These experiments will tell them a great deal about the reef's primary production. They plot a transect or cross-section of the reef, meticulously noting every organism that lies along each link of chain. From the lagoon, past the crest, to the drop-off at the ocean's edge, an accurate picture of reef life will be assembled. The distribution and concentration of plankton and other microscopic organisms are studied. Nitrogen content in algae is compared with nitrogen content in the water. Light intensity is measured at different depths. The team studies the full spectrum, from infrared to ultraviolet, to learn how reefs convert solar energy so efficiently, and why, under some conditions, intense sunlight actually inhibits plant production. Plant samples are gathered from the reef wall some 100 feet down, up to its crest, and into the lagoon. There are more than 100 plant species on this reef. Some are poisonous, while others are favored food for many sea creatures, from parrotfish and sea urchins to snails and crabs. By placing samples at predetermined locations, the team will find out who eats what, when, and how much.
Okay, here's another one. But this is too ragged over on this side to be teeth, so that's probably an urchin as well. There's a good fish. You can see the sort of crescent-shaped bites in there.
At every location, they study the reef's geological history by drilling into the carbonate skeleton to extract core samples dating back thousands of years. But drilling is not always easy. Salt water corrodes the equipment, and if the whole wall collapses, the diamond bit can become irretrievably jammed. There are days when the drilling team wishes it had stayed in bed. Working some 200 miles from the nearest inhabited land can present problems. Repairs must be made with the materials and talent at hand. The Smithsonian expedition is preparing to move to its next location. But first, they make a quick reconnaissance flight to locate other unidentified reefs in the area. The Albatross has helped locate dozens of previously unstudied major reef formations, including ones they are visiting on this expedition.
This ability to draw back and look in a broad kind of way at reef distribution throughout the Caribbean is in fact the very reason why we know that reefs are even there.
The amphibian also serves another very important function: to lead the ship to safe anchorage.
In remote areas in which the terrain is constantly changing, one has to take the hydrographic charts with a grain of salt. The ability to look at the underwater terrain in a broad kind of way allows us to negotiate reef passages that otherwise we would never attempt.
Before journey's end, the team will drop anchor and set up experiments on reefs off the coasts of Haiti, Honduras, Belize, and the Bahamas. At their final location, the marine scientists gather living plant and animal specimens to take back to Washington for further study. The more they learn, the clearer the picture of Caribbean reef life becomes.
The world of natural history is really very complex, and one of the primary jobs of the scientist is to reduce that complexity to something that can be easily understood and worked on. It really is crucial to see enough of the real world to know that the area that he chooses to research as being typical really is typical of that wild world out there.
With each location, questions about a reef's function, its species diversity, its plant productivity, and its history are answered. Studies show that wave action is an important factor in reef plant production. Also, blue-green algae, which are capable of fixing nitrogen from the atmosphere, could account for much of the high degree of productivity in these nutrient-poor waters.
All right, let's hope we're getting some good stuff here.
They now know that a reef's geological history determines its shape and the amount of living coral that will grow on its surface.
Oh, it could be not that much. That has got to be a beautiful, datable piece.
Now
That's
this
a
may be monastery, right?
Yeah, and look at that.
They have learned a great deal, but there are still mysteries that only can be solved at their laboratory in Washington, D.C.
You could go to sea and you could look at the distribution of organisms and you could conclude that, for example, wave action was affecting the distribution of organisms in a certain way. But that didn't prove it. What you really need to do is to bring this problem back into the laboratory where you can control everything.
After nearly 16 weeks among the reefs of the Southwest Caribbean waters, the time has come to relax and prepare for the homeward leg of the journey.
Our captain will lead us through the calm and windy storms. In the Albatross, our loyal friend will fly where we roam. The mornings may find us at a warm, secluded key, where the wind is ever gentle across the beaches to the sea. And sometimes if you're lucky and the current's flowing right, you may see a school of dolphins, such a graceful moving sight. Roll on, Marsus resolute. There ain't no ocean deep enough or wave we cannot shoot. Our captain will lead us through the calms and windy storms. The albatross, our loyal friend, will fly where we roam.
In Washington, the live specimens must be transferred rapidly to their new home at the Smithsonian Institution's National Museum of Natural History. The Living Coral Reef exhibit is the first of its kind, a model of a coral reef community in microcosm. This 3,000-gallon tank is much more than an average aquarium or fish hotel, defying many recent scientific beliefs and attitudes.
It's generally been felt it is impossible to duplicate the marine environment in the laboratory. I had a feeling that it would be possible to bring this community into an aquarium if you were to include the whole community and not a small part of it.
Creating a reef model requires more than gathering plants and animals. The major conditions that affect reef life must also be accurately duplicated.
Reefs are most strongly developed where wave action is quite extensive, and that is one of the elements that we have worked very hard to simulate in our aquarium. One of the major factors with a tropical reef community is sunlight. That was the number one thing to simulate that very intense sunlight.
As in the ocean, a balance of life must be maintained. More than 300 species of plants and animals thriving in this miniature community have been collected and identified. The exhibit's laboratory is set up to monitor and control all factors known to be critical to a reef's integrity. The water is constantly checked to maintain proper levels of oxygen, carbon dioxide, nitrogen, phosphorus, and temperature. The tank has provided masses of new and remarkable information about coral reef communities, but there have been times of frustration.
We thought we had simulated all of the major environmental factors, and yet our community was still marginal in development. Under normal conditions, even at night, there is fresh open ocean water that's driven across the reef to supply oxygen and reduce the levels of toxic excretion of the animals so it doesn't become a problem. But shut the water off and you have a very serious difficulty.
The nearest open ocean water lies hundreds of miles away. In their laboratory, principles learned in the field must be applied to create it. The team decides to make use of a primary anti-pollution device found on wild reefs: algae. They devise the algal turf scrubber, a shallow trough containing a plastic screen to simulate a reef's porous surface. High-intensity lighting and wave action are added. The algae's powers of rapid photosynthesis act as a purifier to produce the equivalent of open ocean water the tank needs when the lights are off.
Once we were able to control the water quality at night, we could do exactly what we wanted to do with the reef. So then we went through a period of about a year of making our reef better than it had ever been before.
The Living Coral Reef exhibit has become a working model of a natural reef, 24 hours a day. There is now time for study and new discoveries.
We had to remove the algal turf, just as happens normally on a reef, with fish are constantly grazing at it, so it could rapidly grow back. Because we were concerned with everything that went in the tank and everything that came out, we would routinely scrape it and weigh the algal turf, and those numbers accumulated. It wasn't until about a year later that we realized that an awful lot of algae was coming off these screens. An awful lot of algae. This left us with the question, why is it that we're able to produce so much algae in such nutrient-poor water? My thought was, well, if we can do it in our artificial reef system, why can't it be done in the open ocean?
To learn if what is happening in the lab really can be made to happen in the wild, the Smithsonian scientists set sail once again for the West Indies. The pilot project is funded by two private non-profit organizations, the Bioenergy Council and Earthwatch. Earthwatch volunteers help the team as sailors, carpenters, and scientific assistants. In a lagoon at the Bahamian island of Miaguana, the 6-month pilot project begins. They build rafts 1 meter square based on the principles of the algal turf scrubbers in Washington. The rafts are placed at varying depths inside the lagoon and in open ocean to find the most productive combination. After weeks of labor and anticipation, the experimental harvest begins.
Okay.
They discover that the rafts are most productive when placed where there is moderate wave action. At depths of 6 to 18 inches, the algae is protected from excessive toxic ultraviolet radiation, but still receives enough sunlight for photosynthesis.
What's giving us our most productivity, what's giving us our most biomass. Susan, do you think you can reach that one corner right there? Okay.
Got the rope, Tim?
I got it. Let me pull her in.
Okay.
Okay.
As time passes and accumulated data mounts, the evidence becomes overwhelming. Plant production can exceed any previously thought possible.
We had predicted it, and we really expected it, but our minds were so ingrained that this could not be so. It was amazing to us that month after month, these screens were producing at the rates of 5 to 10 times the best Midwestern agriculture.
Their experiments make it clear that these tiny aquatic plants could be an answer to the world's nagging problem of maintaining adequate food production. The question now becomes, how can we best make use of this extraordinary potential?
Perhaps what we really should be doing is searching for a grazer which could feed upon this algae.
They experiment with numerous reef grazers, including queen conch, whelk, and parrotfish, before settling on an elusive large spider crab, the mithrax, known as the Caribbean king crab.
We knew it in the wild as a very large crab, as big in mass as the famous Alaskan king crab, but it wasn't a common food because Caribbean fishermen had difficulty in catching them. We decided to see if mithrax could be domesticated. Unfortunately, we never realized how difficult it would be to find the crab in such numbers that we could experiment to see if this could be done.
It takes 3 months searching high and low to locate enough of these shy and evasive crabs to learn their habits. They discover that the mithrax hides in four-reef caves, only coming out in numbers at night to feed. The puzzle of the mysterious mithrax is solved.
Here we had the perfect sea cow, if you will, on which to feed our algae. That's a good-sized man.
But there are still many steps in the process of domesticating this Caribbean king crab.
Our next problem was, how were we going to hatch Mithrax crabs from eggs? And this could have been a very serious problem because a large number of marine organisms, including most crabs, have an initial planktonic stage maybe as long as several months in open water. As it turned out, nothing could be simpler than the Mithrax because the planktonic or open ocean stage is about 24 hours long, and then it settles down as a small megalops stage, it's called, which immediately starts feeding on algae. So we have all of the pieces now to make massive animal production in a single step. The Mithrax is really not far from the Alaskan king crab, one of the biggest crabs, and yet it eats the smallest benthic algae. In my mind, we had brought together all of the elements, but it was necessary to demonstrate that you could do it by the hundreds, repeatedly, whenever you wanted to do it.
With support from the Agency for International Development, the Peace Corps, and Department of State, the Marine Systems Laboratory begins an experimental project at the British colony of Turks and Caicos. They develop a commercial fishery based on what they have learned. The program is expanding to several countries throughout the Caribbean basin, including the Dominican Republic and Antigua. Combining intensive field work with modeling techniques in the lab, this unique Smithsonian team of scientists has developed concepts and procedures that could greatly increase our world's food resources. In an attempt to better understand these complex and exotic biological communities, what began simply as scientific curiosity may ultimately benefit us all.